Japan is investing a lot into longevity research in hopes of keeping us young forever. And recently, they managed to bring about a new kind of vaccine which manages to stop the aging process very reliably through gene editing and biotechnology. This research team intends to move to human trials very soon. Want to know about it? Well, stay tuned to the video and watch the video till the end. Hello guys, welcome to Futureality. Hit the subscribe button and turn on the notifications to get new updates about technology and development. Hit the like button to show your support, and in today's video, we're going to discuss is Japan's anti-aging vaccine the future of longevity? So without any further ado, let's begin. The world has seen how quickly a vaccine can be developed and rolled out when there is an urgent need. Now is the time for researcher and big pharma to bend their resources in the direction of vaccines for aging. Imagine how many age-related diseases could be ameliorated if we could vaccinate against renaissance, and the enormous benefit not only for people but for healthcare systems, the economy, and the whole of society. For the new study, researchers in Japan set out to find a way to target senescent cells more directly while leaving healthy cells alone. By examining gene expression in senescent cells, the team first identified a protein called GPNMB, which is expressed in high levels by these defunct cells. This protein was also detected in high levels in patients with atherosclerosis, which is linked to senescence. But the big question is whether that vaccine would be safe in humans. Robbins, who was not involved in the research, told Life Science. To figure that out, the researchers would need to conduct additional studies in animals, including non-human primates, and thorough safety trials in human patients. Cellular senescence refers to a state of stable cell cycle arrest in which proliferating cells become resistant to growth-promoting stimuli, typically in response to DNA damage. The terms aging and cellular senescence cannot be used interchangeably. Aging is a progressive decline with time, whereas senescence occurs throughout the lifespan, including during embryogenesis. The number of senescent cells increases with age, but senescence also plays an important role during development as well as during wound healing. They're asking the question of what's the longest life that could be lived by a human complex system if everything else went really well and it's in a stressor-free environment, says Heather Whitson, director of the Duke University Center for the Study of Aging and Human Development, who was not involved in the paper. The team's results point to an underlying pace of aging that sets the limits on lifespan, she says. The physiological importance of cell senescence has been attributed to the prevention of carcinogenesis and, more recently, aging, development, and tissue repair. Senescent cells contribute to the aging phenotype, including frailty syndrome, sarcopenia, and aging-associated diseases. Senescent cells secrete a series of factors that disrupt the function of the tissue. Dr. Salvador Makib, head of the University of Leicester's Mechanisms of Cancer and Aging Lab, told Euronews Next. They call cells from the immune system, in theory, to be cleared by them, but that eventually fails, and create a chronic low-level inflammation mixed with fibrosis, Makib said. Makib was part of an international team of academics from universities in the UK, Spain, Nigeria, and Saudi Arabia that published research on another method of tackling senescent cells. The biological process of aging is very complex, therefore it's unlikely that one single strategy will completely stop it or reverse it. However, there are probably many ways to slow it down, and clearing senescent cells seems to be one of the easiest and potentially more effective, he said. We can expect that the vaccine will be applied to the treatment of arterial stiffening, diabetes, and other aging-related diseases, Minamino said. The results of the team's research were published in the online version of the journal Nature Aging. Senescent cells refer to those who have stopped dividing but do not die. They damage nearby healthy cells by releasing chemicals that cause inflammation. SASP-produced chemicals adversely affect neighboring cells, the surrounding extracellular matrix, and other structural components, causing a cascade of negative effects, which can include chronic inflammation, arthritis, and arterial stiffening, as well as triggering senescence in healthy cells. Cells cannot keep dividing forever. Eventually, they accumulate too much DNA damage through environmental stress. So the body shuts them down and flags them to be cleared out by the immune system. This seems to be an evolutionary defense mechanism against cells turning cancerous. However, even immune cells aren't immune from this process, and as they become senescent themselves, the immune system gradually loses the ability to clear out senescent cells. As these inactive cells accumulate in the body, they contribute to symptoms of aging and the diseases that come with it. The potential benefit of using a vaccine rather than drugs to target senescent cells is that people could be given the shot at, say, age 50 and avoid accruing senescent cells in the first place, Robin said. A vaccinated person's immune system would be trained to look for senescent cells and destroy them on sight. In contrast, someone taking senolytic drugs would need to take them repeatedly, as senescent cells would reaccumulate after each course of treatment, he said. Measurements such as blood pressure and blood cell counts have a known healthy range. However, Whitson points out, whereas step counts are highly personal, the fact that Pycroft and his colleagues chose a variable that is so different from blood counts and still discovered the same decline over time may suggest a real pace of aging factor in play across different domains. Study co-author Peter Fejdchev, who trained as a physicist and co-founded Giro, says that although most biologists would view blood cell counts and step counts as pretty different, the fact that both sources paint exactly the same future suggests that this pace of aging component is real. By modifying just one gene, an international team of researchers was able to extend the life expectancy of mice by 23%, and they think their results may translate to humans. Every day is a day closer to technological singularity, experienced robots learning to walk and think, humans flying to Mars, and us finally merging with technology itself. This is a very interesting question, and one that we still have not agreed upon. Some believe there's a hard limit for human lifespan, around 130 years is the current estimate, while others think that on paper, immortality should be feasible, Makib said. It's still early to know how much life can be extended and whether there's a limit or not. The field of anti-aging research is advancing very fast. In the last decade, there have been many key discoveries, Makib said. The person that will take the first anti-aging pill has probably already been born. Using an amino acid, in GPNMB, the research team created a peptide vaccine which enabled the body to create antibodies that attach themselves to senescent cells. Once attached, leukocytes finish the job by attaching to the antibodies and removing the senescent cells. This was then tested in 3 groups: young mice on a high-fat diet, middle-aged mice, and mice with an accelerated aging disease known as progeria. Improvements were seen in all 3 groups. The high-fat diet mice had better metabolic function than mice on the same diet that didn't receive the vaccine. Middle-aged mice that were vaccinated at 50 weeks of age remained more active and had faster movements by 70 weeks than controlled mice. Vaccinated mice with progeria had a significantly longer median lifespan than unvaccinated animals, with the effect more pronounced in male mice. Evidence suggests that molecules spewed by senescent cells directly contribute to the buildup of plaque in atherosclerosis, according to a 2020 report in the journal Clinical Chemistry and Laboratory Medicine. With this in mind, the team wanted to see whether expunging GPNMB heavy cells from the body would help reduce the density of these plaques. Our study has demonstrated the possibility of a new anti-senescent strategy, said Professor Tohru Minamino, corresponding author of the study. We speculate that there are many more xenoantigens that are produced by other kinds of senescent cells. With more research, we'll be able to provide individualized anti-senescence therapy for patients depending on the prevalence of different types of senescent cells in their body. Now that we've come to the end of this video, I want to thank you for sticking with me, and I'd love to know what you think of it. Just comment down below. Also, if you like this video, make sure you like it and stay safe. Today's video is over, but if you want to see more, there's one on your screen right now, and there are a few more fun videos coming soon. I'll see you in the next video.